So we'll discuss about centrosome. So centrosome is an organelle which is found only in animal cells. So that is a very important point. It is found only in animal cells, not in plant cells. So centrosome is something which is found only in animal cells. So this is found only in animal cells. Also, we have discussed earlier that centrosomes are also non-membrane bound organelles like ribosomes. So these are examples of two cell organelles which are non-membrane bound. So that is another important feature. This is also non-membrane bound. So if you're asked to choose two cell organelles which are non-membrane bound, you can write either ribosome or centrosome. So centrosome, it contains two cylindrical structures inside. These two cylindrical structures are called centrioles. So centrioles are cylindrical structures which are found inside the centrosome. So I'll show you a picture. So you can see here, these are the two centrioles, and you can see here they are usually arranged somewhat perpendicular to each other. So if this is a centrosome, assume this is a centrosome, then you'll find two centrioles inside which are arranged somewhat parallel to each other. So assume this is one centriole, there will be another centriole here, and they are arranged almost perpendicular at 90 degrees to each other. Okay, so centrioles are structures which are found inside the cell organelle called centrosome, and there are two centrioles in one centrosome. These centrioles, they are surrounded by amorphous pericentriolar materials. So they are surrounded by amorphous pericentriolar materials. The word amorphous means non-crystalline, which is not crystalline in nature. They are solids but not crystalline in nature. So we call this as PCM, amorphous PCM. PCM refers to pericentriolar material. So pericentriolar material or pericentriolar matrix is a mass of protein which surrounds the two centrioles in a centrosome. So I'll just mark it here. So something surrounding these centrioles, this amorphous mass of proteins is referred to as amorphous mass of proteins is called pericentriolar materials. So we'll just label the parts here. So in this diagram, this represents the centriole, and the yellow color portion represents, so this is a centriole. This yellow colored portion, it represents the PCM, pericentriolar material. Okay, so pericentriolar material is a mass of amorphous material that is found surrounding the two centrioles. And like I mentioned earlier, the centrioles, they lie perpendicular to each other, and they have an organization like the cartwheel, which we'll discuss later. Why is it called a cartwheel-like structure? Now, the structure of centriole is almost similar to the structure of Celian flagella. In Celian flagella, the structure was 9+2. In this case also, it is somewhat similar to that, but there are some differences also. So we have discussed earlier, in Celian flagella, there are 9 peripheral microtubules and a pair of central microtubules. And these peripheral microtubules are doublets because there are 2 rings. But in this case, it also has 9 peripheral fibrils, which are made up of microtubules or tubulin protein. But in this case, it is not a doublet. In this case, it is a triplet. So instead of 2 rings, it will be having 3 rings. So this is the first ring, this is the second ring, and this is the third ring. So A, B, C. So the peripheral microtubules are going to be a triplet instead of a doublet, and there will be 9 of them. So instead of 3 rings, they'll be made up of instead of 2 rings, they'll be made up of 3 rings of microtubules, A, B, and C. So A has around 13 microtubules. B and C has less than 13 because they share some of the microtubules. Okay. So yes.
Sir, can you explain that once again? So I didn't understand.
Which one?
So that microtubules.
Okay, 1 second.
So I just want to ask that where are they in that centrosome?
these centrioles. Are you asking about centrioles?
Yes, sir.
No, you were asking where are they, right? What they as in what?
Centrioles.
Centrioles just lie randomly inside the centriole, but they'll be just perpendicular to each other. So centriole is made up of only two things. It has this yellow colored portion called pericentriolar material and this blue colored things, this thing. See, these are the two centrioles, and if you put this in a circle, then that is called a centrosome.
Centrosome.
The centrosome contains two structures called centriole, and surrounding this centrioles, you'll find structures called pericentriolar material. That's it. Now, if I take this centriole and cut it, then you'll find structures like this. So if you look here, you can see here there are three. This is a triplet. This is a triplet. This is a triplet. Okay. So these
Yes, sir.
centrioles will be made up of microtubules.
Okay, sir.
Okay. So let's discuss again. So if right now I'm discussing the structure of each of these centrioles, if you look in this picture, you can see here, this much is one triplet here. Then this is another triplet. You can see three microtubules, another one here. Okay.
Yes.
So yeah, instead of doublet, in case of Celian flagella, it was a doublet, but in this case, it is going to be triplet. Similar to Celian flagella, in Celian flagella, the doublets were also linked. Similar to that, in this case also, the adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and also called the hub. So if you remember the structure of Celian flagella had two microtubules in the center. We call them as central microtubules, and 9 doublets in the peripheral region. It was somewhat like this. So 9 of them were there. Somewhat like this, and these were also linked to form a circular structure. And this was referred to as the 9+2 structure. Now, in this case, there is no central microtubule. Instead of central microtubule, it has a proteinaceous rod-like structure, cylindrical structure in the center. This proteinaceous rod-shaped or cylindrical structure in the center is referred to as the hub. Okay, so in this case, there is no central microtubule. There is only a proteinaceous rod-like or cylindrical structure called the tube-like structure called the hub. So you can see here in this diagram, this central portion, this red colored circle in the center, this one, this is called the hub. So hub is not a microtubule, it is a proteinaceous rod-like structure. It's called the central hub. So surrounding the central hub, you can see here, there are 9 triplets. So 9 triplets, you can see here A, B, C. So subfiber A, it is complete with around 13 protofilaments or microtubules, but B and C, they are incomplete because they are sharing some of the microfilaments. And the adjacent triplet fibers, you can see they are connected by some linkers. These are called the linkers. Also, the central rod is connected to these peripheral microtubules by means of a spoke. This is called the spoke. So total, how many spokes will be there? There will be around 9 spokes because there are 9 peripheral microtubules. And this is called the linker, AC linker, because it links A of 1. So if this is A, then this will be C. A, B, C, A, B, C. It is called AC linker because it links A of one microtubule to C of another microtubule triplet. Okay, so this is a structure of a centriole. Now, if you look at the structure, it looks somewhat similar to the wheel of a cart. That is why the structure is also known as cart wheel structure. So because the structure has a resemblance to the wheel of a cart, this is also called cart wheel structure. Okay, so the hub is connected with the tubules of peripheral triplets by radial spokes, which are also made up of proteins. Now, can we call this structure as 9+2? similar to what we did for cilia and flagella. Can we call this as 9 plus 2? Kedar?
So how come 2? So they are only 9.
Okay, then instead of 9 plus 2, what can you write instead of 9 plus 2?
So only 9.
Okay, so we can write 9 plus 0 because there's nothing in the center, no microtubules in the center. There is only a proteinaceous hub in the center. So this arrangement is called 9 plus 0 arrangement. So always remember that 9 plus 0 arrangement is found in centrioles. 9 plus 2 arrangement is found in Celian flagella. So this is a bit confusing, but very easy. So 9 plus 2 is in Celian flagella because they have 2 central microtubules. But in centrioles, they don't have any central microtubules. So that is why it is 9 plus 0. They have only something called hub. So hub is a proteinaceous. It is made up of proteins and it is found in the center. Last point, the centrioles form the basal body of cilia and flagella. So that point is important. So cilia and flagella, they are long structures. They should have something called a basal body. So basal body is a portion of cilia and flagella that is inserted in the cell membrane or the cell wall. So this basal body is actually, it arises from the centrioles. It is made up of centrioles. So that point is very important because this was asked once in NEET exam. So centrioles form the basal body of cilia and flagella. The most important function of centriole is, of course, cell division. So centrioles help in the formation of spindle fibers in cell division. You will study in detail about spindle fibers in chapter 10. There is an entire chapter about cell division and cell cycle. So spindle fibers, I'll just give you a summary of what spindle fibers are. If you're from ICSE, you might have already studied about cell division. But CBSE students, they don't have this topic in 9th and 10th. So I'll just mention what this is. So during cell division, chromosomes will be like this. So this is one chromosome. This is another chromosome. So chromosomes will align at the center. Now, during cell division, you know that these chromosomes should equally pass to each of the daughter cells. If it has to equally, if you have to divide this equally to each of the daughter cells, you should pull these chromosomes from both the sides. Okay. So if you want to pull these chromosomes from both the sides, you should attach something to this. So you can attach some fibrous structure like a rope-like structure here and start pulling from both the sides. So this fibrous structure, which attaches to the chromosome during cell division and helps separation of the chromosome, is called spindle fiber. So you'll understand this better when we study cell division chapter. So right now, just remember that spindle fibers are fibrous or thread-like structures which attaches to the chromosomes during cell division. It splits the chromosome into two so that it can be equally divided among two cells. And these spindle fibers are formed from structures called centrioles. So without centrioles, spindle fibers cannot be formed. And this is very important in case of cell division in animal cells.